Hello everyone, my name is Gary O'Mealy, and in this video we are going to discuss phospholipids and the cell membrane. We've talked about the cell membrane before in how it acts as a barrier that helps to separate microscopic compartments in the body. The specific example we provided for this is that the cell membrane separates the intracellular fluid, or the fluid found inside of cells, from any of the extracellular fluids, which include the interstitial fluid, blood plasma, lymph, and other examples like that. So we haven't really talked much about the actual composition of the cell membrane, so we're going to go ahead and start talking about one of the major components, which are phospholipids. We also introduced phospholipids in our video on lipid structure and function, but we didn't say a whole lot about that because we knew we were going to talk about it in this video here. So quickly, let's review phospholipid structure. First, you have a glycerol backbone. Attached to one of the three hydroxyl groups on glycerol is a saturated fatty acid. Another of the components of glycerol is taken up with an unsaturated fatty acid, and you'll recall that the presence of a cis double bond in the unsaturated fatty acid hydrocarbon causes the tail to kink out a little bit, causing this tail to take up a little bit of extra room, giving the phospholipids some lower density, and that's going to become important here in just a couple of minutes. So a phospholipid is a diglyceride in that the glycerol, which can bind up to 3 fatty acids to become a triglyceride, instead only binds 2. The third position that would otherwise be taken up with a fatty acid is taken up with a negatively charged phosphate functional group, which is attached to R. And R, similar to what we saw with amino acid side chains, refers to the remainder of the molecule. R can refer to choline, ethanolamine, serine, inositol, or several other examples. So there are several different types of phospholipids that we can find in the membrane, and they are differentiated based largely on what that R group is. So this creates a very interesting situation for a phospholipid, especially since we are used to, at this point, the idea that lipids in general are very hydrophobic. Rather than being entirely hydrophobic, the phospholipid is going to have one portion of the molecule which consists of a very large number of polar functional groups, and we consider this to be the polar hydrophilic head of the phospholipid. And then more of what you would expect down here, which are the nonpolar hydrophobic tails. So this is a very interesting situation in which this is a lipid, which again we would normally expect to be almost entirely hydrophobic. Half of it is hydrophobic as we would expect, but the other half is actually very hydrophilic and interacts very well with water. So phospholipids are what we call amphipathic molecules, meaning that they have both hydrophilic and hydrophobic properties. So if we're going to get into the business of designing a cell membrane around using these phospholipids, we need to think about how best to make use of these properties. And what we're going to see is that when you actually construct a cell membrane out of phospholipids, the best way to do it is to create what is called a bilayer, in which you have 2 layers of phospholipids stacked on top of each other and flipped upside down, so that the hydrophilic heads, which are labeled here on yellow, are always pointed out towards the water that are in the various bodily fluids. You can see that the extracellular fluid up here, which has water in it obviously, is exposed to the hydrophilic heads on this leaflet that is pointing upward, and the water that is in the intracellular fluid is pointed towards the phospholipid polar heads that are pointed downwards from that bottom leaflet. What that leaves is the non-polar hydrophilic tails are pointed inward at each other, and they create what is called a hydrophobic core in the membrane, which is a portion of the membrane that almost completely excludes water and any other polar hydrophilic substances. So creating a bilayer like this is clearly the best and most optimal way for phospholipids to construct a membrane that has a high surface area. So you can kind of imagine that if you decided to try to create a phospholipid membrane that only consists of one layer, The polar heads would be pointed towards water, which they would like, but you would have to have your hydrophobic tails pointed towards water as well, which they would not like. So having a bilayer like this satisfies the hydrophilic heads in both cases because they're both pointed towards water that's either in the extracellular or intracellular fluid, and then the hydrophobic tails are happy because they are pointed inward at each other and they never have to see the water that is in either of the bodily fluids. So again, the hydrophilic heads point towards water. That's a good thing. Hydrophilic means you like water. And the hydrophobic tails point inwards and they exclude water. That is also a good thing in this case because hydrophobic, again, means water-fearing. They do not want to see water, and based on the way we are constructing a phospholipid bilayer, they don't have to see water. So other than phospholipids, we're going to talk about some of the other components of the cell membrane. There are 4 major components of the bilayer, of which we have already talked about phospholipids. We're going to talk a little bit about cholesterol that wedges itself into the membrane. We briefly talked about this in our video on lipids. There are proteins that wedge themselves into the cell membrane, and then there are carbohydrate chains that we often find on the cell membrane as well. So in order to fully understand the role that cholesterol plays in the cell membrane, we need to talk about the membrane's freezing point. Now, I don't want you to think necessarily of the membrane turning to ice necessarily. Think of it more as the membrane solidifying. The ability of the cell to transport materials through the membrane is owed largely to the fact that the membrane has a lot of fluid-like properties. If the membrane were to solidify, meaning that it turns into a solid, then membrane transport would no longer be able to happen. Try to run through a brick wall. See how much success you're going to have getting through the brick wall. Well, that's because the brick wall is a solid. If the brick wall were more fluid, you might have a chance of getting through. So let's talk about the role that cholesterol plays in maintaining membrane fluidity. So let's consider a couple of situations here. In the first situation, we have no cholesterol in the membrane, and the fatty acids that are attached to the phospholipid heads are going to be saturated fatty acids, So if the phospholipids have nothing but saturated fatty acids, then those tails are going to stick straight out, and because there's no cholesterol in here, these phospholipids are going to pack together very tightly. They're going to have a very high density, and this is going to produce a membrane that is very likely to freeze or solidify at normal body temperature, which is 37 degrees Celsius. However, thankfully for us, this is not what our phospholipids look like in the membrane. Rather, our phospholipids look a little bit more like this. There is cholesterol that wedges itself into the hydrophobic core of the membrane, forcing those phospholipids to space themselves out a little bit further. You do have the presence of unsaturated fatty acids, which again have these kinks that again cause the phospholipids to space themselves out a little bit more. And this results in a more low-density membrane, and at a lower density, the cell membrane is a lot more unlikely to freeze or solidify at normal body temperature. This keeps the membrane in much more of a fluid-like state, allowing for membrane transport processes, which we will talk about very soon in future videos, to become a lot more efficient. So this situation right here is much more optimal for biology and biological processes, and thankfully, this is also the reality of the situation. Okay, so now let's talk about membrane proteins. There are all sorts of membranes that you can find wedged in the membrane, and there are proteins with a number of different very important functions. We're not going to talk about all of them here in this video, but we will highlight a couple that are going to become very important in numerous physiological processes. First, you have what are called transport proteins. Transport proteins allow for the passage of ions and molecules through the cell membrane. You can see one of them right here. This one is called a channel protein. You'll notice that there is a hollow portion that goes entirely through the length of the membrane, and what this allows for is that if you have a molecule that otherwise would not be able to pass through the membrane, it is allowed passage to go straight through like that, to go from one side of the membrane to another. Then on the other hand, you have receptor proteins. Receptor proteins allow a cell to receive and respond to signals from elsewhere in the body. We talked about this sort of thing all the way back in our very first video on homeostasis. We've talked about the role that neurotransmitters and hormones play in conveying information over long distances in the body. These receptor proteins allow effector cells to receive those hormonal or neural signals from the cells that are sending them. So this red square here is going to be some kind of signaling molecule, whether it be a hormone or a neurotransmitter, and when it binds to its receptor, as you can see here, that is going to cause the cell that has the receptor to produce some kind of physiological response. And then enzymes you often find embedded in the cell membrane. We've obviously talked ad nauseum about enzymes already. Enzymes embedded in the cell membrane can help produce a molecular response to a signal. So you can see here a substrate that is available in the intracellular fluid. When this enzyme that's in the membrane becomes activated, it can start converting it into product. And it's no coincidence that I drew the enzyme being located in very close proximity to the receptor protein, because receptor proteins and enzymes often end up being linked together, so that you end up in situations where the signaling molecule binds to the receptor, and this somehow leads to the activation of the enzyme, and the production of product is a big part of whatever the physiological response that we desire is. Okay, so keeping the subject here, as far as membrane proteins go, there are two basic varieties. Integral membrane proteins span the entire width of the membrane. The channel protein on the left and the receptor protein on the right are good examples of this. Whereas the enzyme that you see here, which associates with only one leaflet of the membrane and not the other, is what we would consider to be a peripheral membrane protein. Both integral and peripheral membrane proteins are very important in a number of different physiological scenarios, and we will see plenty of examples of them as we go throughout the course. Okay, so since we are just on the tail end of our discussion on protein composition and protein folding, I wanted to bring that discussion full circle to talk about the actual folding and composition of a channel protein. We saw before that a channel is an integral membrane protein that allows the passage of molecules from one side of the membrane to another. These channels are sometimes called water-filled channels because not only are you going to have water on either side of the membrane, but if we're allowing for a hollow channel to connect the ICF and the ECF, that channel is going to be filled with water as well. So we're going to need to take what we know about how proteins fold and how they interact with the water that's around them and apply it to this situation here. So a channel protein is going to fold so that any hydrophilic amino acid side chains will project into the hollow water-filled portion of the channel, as you can see here. So any places where we find water molecules, you can see that the water is facing the green portions on the surface of the folded structure of the channel. Those are going to be the hydrophilic side chains like serine and threonine and glutamine, and maybe some acidic or basic ones like aspartate, glutamate, lysine, arginine. And then the hydrophobic side chains are going to interact with the actual hydrophobic core of the channel, as you see here. These side chains are interacting with the fatty acid tails of the phospholipid bilayer. So again, we can see that a folded structure of a protein, the protein folds in such a way that all or as many of the amino acid side chains are as satisfied as they possibly can be. Okay, and then the last component of the cell membrane that we want to talk about are glycoproteins and glycolipids. A glycoprotein is just a regular folded protein that has been chemically modified with carbohydrate chains. Similarly, a glycolipid is a lipid that has been chemically modified with carbohydrate chains. So in this diagram, you can see not only have some of the phospholipids been modified with some polysaccharide molecules, but so has this receptor protein here as well. The attachment of carbohydrate chains can, in sometimes subtle but very important ways, modulate the activity or function of phospholipids or of proteins. So the important thing I want you to take away from our discussion on glycoproteins and glycolipids is actually a very simple one. These carbohydrate chains are only ever found on the surface of the membrane that faces towards the extracellular fluid. You will never, ever, ever find these carbohydrate chains pointed towards the inside of the cell or pointed towards the intracellular fluid. Doing it this way allows for the cell membrane to have some polarity to it and allows the cell to always be able to tell which side of the membrane is which. And importantly, and we will have a discussion on this in the future when we talk about blood typing, the presence of these carbohydrate chains that face towards the outside of the cell, they help to participate in the body and the immune system's ability to recognize cells that are indigenous to the body and differentiate them from cells that are from outside sources, whether it be from blood donations, tissue donations, organ transplants, or if you're infected with bacteria, the body needs a way to distinguish native cells from invading cells, and these carbohydrate chains help the body to do that. Okay, that's going to do it for this video. Here is a list of vocabulary terms you probably are going to want to be aware of. And then for checking your understanding, number 1, describe the composition and properties of a phospholipid and why does it require that the membrane be a bilayer. Number 2, why does the membrane need fluidity and what helps to maintain this? And number 3, what roles do membrane proteins play in cell function? Okay, that's going to do it for this video. If you have any questions, go ahead and leave them in the comments section, and I will see you for the next video.